The Hokkaido wolf is an extinct species that used to live within the forests of Japan. Due to the long lack of these predators, there has been a major imbalance of biodiversity, which causes issues within the habitats and environments. With fairly recent scientific breakthroughs, there's only one factor that can help solve this. Take it away. Yeah.
Thank you very much, ladies and gentlemen. Right now, I got to tell you about the fabulous, most groovy gene editing. Gene editing. Yeah! Gene editing. Gene editing. Gene editing. Gene editing. Gene editing truly make me wanna clone.
Yikes. Hi, I'm Alexander Abe, and what you just saw was supposed to grab your attention. And it worked, right? My job here in this video is to teach you about using gene editing to de-extinct the Hokkaido wolf, and why we should do it. But before we get into the Hokkaido's who's, what's, where's, and why's, you're gonna need some background information, and quite a bit of it too. So unless you already know CRISPR Cas9, mitosis, meiosis, fertilization, and how to clone a living creature, then I suggest you stick around. Without further ado, let's get right into it. We'll start with CRISPR Cas9, so here we go.
CRISPR Cas9 is a tool used for editing genes that was discovered by Jennifer Doudna, Emmanuel Charpentier, and Fed Zhang. I apologize if I am completely slaughtering those pronunciations, but let's move on. This tool is able to help us with a number of things when it comes to de-extinction. CRISPR Cas9 can repair a sequence that may have decayed over time from a piece of an extinct species. It can remove mutations or genetic diseases, and even make an animal have traits that help it adapt to an environment. It can be used to do all of these things by having the ability to carefully cut genes out of a DNA sequence and use programmed guide RNA to edit that selected gene. But before we knew the capabilities of CRISPR-Cas9, the discovery of CRISPR itself had to occur. In nature, some bacteria immune systems use CRISPR to protect from viruses. It does this by copying the genetic code of the virus and sending it to the immune system so it can use that information to fight the virus. CRISPR was discovered by Francis Mojica, who is a professor at University of Alicante in Spain. He discovered CRISPR while studying halophilic microorganisms, which are organisms that enjoy high salt environments, when he found a peculiar section of regularly spaced repeats, which essentially is CRISPR, CRISPR standing for Clustered Regularly Interspaced Short Palindromic Repeats. He and his research group then found its natural function while studying E. coli in the early 2000s.
Wow, that CRISPR-Cas9 sure is pretty neato. Now, let's learn about mitosis, meiosis, and fertilization. Check it out.
Here's a diagram I made to teach you about mitosis, meiosis, and fertilization. This blue blob represents a somatic cell, and within the somatic cell, you can see there's something called a centriole. Now, I'm not going to go too much into it, but it basically organizes and structures everything within the cell. There's also a nucleus, and within the nucleus there's DNA, which is the code for the organism that this somatic cell belongs to. In the first stage of mitosis, the DNA copies itself within the nucleus membrane. This is called interphase. Then, the DNA wraps itself into chromosomes, and those chromosomes separate onto either side of the somatic cell. These are called the prophase and metaphase. Finally, the somatic cell splits into two somatic cells, and the chromosomes unwind. These are called anaphase and telophase. Then, in meiosis, the The DNA wraps itself in the chromosomes. It doesn't copy itself. It wraps itself and then they separate. And the somatic cells split again, but this time with only half as many chromosomes as they had before. These are called gametes. Then, in fertilization, another gamete from the opposite sex comes together with the other gamete to form a zygote. This performs mitosis again to create a multicellular organism. This is mitosis, meiosis, and fertilization.
Wow, that was some pretty interesting stuff. Now, let's learn about how to clone any living creature. Let's go.
This is how you clone an animal. First, get some DNA from a preserved fossil of your extinct species and place it inside a non-DNA containing egg cell from a distant relative of the species. In the Hokkaido wolf's case, it's the gray wolf. Then, add a small electric current to force your somatic cell to undergo mitosis so an embryo forms. This embryo is then put into a surrogate mother for it to grow and finally be birthed as a close clone of the original extinct species, the Hokkaido wolf. Oh, and it's important to mention that if there is by any chance a genetic mutation and people are worried about it spreading, well, the Hokkaido wolf is on an island, of an island, so there's really no risk there. Anyways, that's all I have to say.
Well, now that you have all of that scientific information locked in your noggin, it's time to get into the fun part. I'm so excited about teaching you the history of the Hokkaido wolf and why we should consider bringing it back that I wrote a little song about it.
In late 1860s Japan, the Meiji Restoration began. Farmers started ranching, agriculture was advancing, but one thing stood in the way of their plan. The Hokkaido wolf was the predator, and livestock was its prey. The government killed all of them, but didn't know the price it would pay. Over the years, invasive deer caused forests to disappear due to the amount of predators that were mere. Just like in Yellowstone National Park, biodiversity was also slim in this landmark. So scientists decided to reinvite a wild canine with the thought that it would make biodiversity fine, and it did. It worked. The farmers weren't too happy, but to be honest, it was kind of the wolves' territory first. Now this brought up a solution for the problem at hand. If it worked in the past, then it should work with Japan. But since the Hokkaido wolf is extinct, there'll be a few extra steps. We use CRISPR Cas9, the gene editing tech. We gotta get at the wolf's DNA, edit it if it's decayed, put it in a relative's donor egg, then send it off to where it once preyed. Deer populations would be fewer. But how is this best for the future? In 1998, hunting regulations were low, and there was a loss of about 72,000 bucks and does, but still to this day they threaten the forests of Japan, so here's a clear solution that I have to propose. We de-extinct the wolf, make sure it's well and alive, then similar to Yellowstone in 1995, we bring it back in small groups and allow it to thrive, and if it shows to work, then the forest will revive. But if this plan fails and the imbalance doesn't end, then we'll make like 1889 and kill them all again. It's that easy.
Well, now that you've heard my proposed solution and my sick beats, well, I don't really have that much else to tell you. I hope I've left you with more knowledge than what you came here with. Unless you're one of the 50 or so students who also did this project with me and just skipped straight to the rap part, which I don't blame you. That's anyways, I thank you for watching, and good night. Or should I say, good morning.